Yes.
Perfect. So let me begin. Good evening, guys. And first of all, I would like to thank Thai Toronto for having us again on this platform, this wonderful platform, and presenting or introducing Enrich Bioscience. Let me begin by saying that we all share a common story. We lost someone, a family member, friend, or relative from cancer. Better cancer treatment outcomes demand early detection. Enrich Bioscience is developing an early detection test that is accurate to you and your loved ones. My name is Tarang Khare, and I am the founder of Enrich Bioscience. I am Indian by origin, South Asian descent, and by training, a biomedical scientist. During my research, I learned that cancer biomarkers behave differently on different races and ethnicity. In other words, most molecular tests available to my family and for me in North America may be inaccurate. And the reason is that most studies and molecular tests are developed on European descendant. To illustrate on this, let me share our preliminary results from an ongoing retrospective study in collaboration with Dr. Satyamurthy in India. On the y-axis is the accuracy of the test model, while the x-axis represents different ancestry. We trained cancer models on European descendants and applied them to other lineages. And what we observed? A drop in accuracy of more than 20% across various origins. Other investigators are also observing the similar findings on ancestry disparity with cancer models. There is an unmet need to develop cancer models for non European patients. Cancer diagnostic is a crowded space, and most companies are in North America and Europe, targeting patients of European descent. Enrich Bioscience wants to change this. We want to develop cancer models for non European patients. And our first market is India, and it is for the following reason. India has the highest number of cancer deaths among new cases, 26 deaths among every 100 new patients, while for the US and Canada, this number is 8. Second, Indian patients are underrepresented in global cancer genome studies, and we want to bring Enrich Bioscience data on Indian patients, which will add value to the current scientific knowledge. And the pertinent aspect is that our partners in India see value in Enrich Biosciences approach, and they have offered their services at a moderate price, and it makes the cost of the product development pipeline 10 times less compared to the North America. However, the bottleneck is that India is a price sensitive market. The market demands for an affordable molecular testing. To that, we developed a cost effective testing workflow. The test itself analyzes thousands of biomarkers to detect various cancer from the single blood specimen. Enrich Bioscience technology is built upon the science of DNA methylation. It is the modification on the core of the sequence, and its patterns are unique for each tissue and cancer type, and therefore it offers high specificity for cancer detection. And our two step test design is a good fit to address the market's price sensitivity. Step 1 informs if cancer is present and growing in the body, and if the test results are positive, then only we are proceeding to step 2 to identify what type of cancer and to characterize the cancer, including the stage. So, NH Bioscience method is 100 times more sensitive and 4 times faster than the closest academic protocol. And we have developed a deep learning algorithm for the robust detection of cancer markers on background noise. Let me elaborate more on the AI component of Enrich Bioscience test. Step 2 of our test generates a pattern from thousands of biomarkers associated with a cancer type from the blood sample. And working with the blood sample is challenging since cancer originated material is in minuscule amount and they results in incomplete data generation. And shown here is an analogy between the biomarker patterns and the famous Mona Lisa painting. The picture gets distorted when the data points are missing. And similarly, the AI model accuracy suffers from the missing data points. And so, what we have done, we have developed a deep learning algorithm that can tolerate up to 40% of missing data and still can detect cancer types with more than 96% accuracy. Oops. Enrich Bioscience, oh, Enrich Bioscience has a strong scientific team backed by scientific advisors from Canada, the US, and India. We are excited and honored to have an ongoing partnership with distinguished oncologists from India. We have strategic partnership in place with Tata Medical Center, Calcutta, and AIMS, New Delhi, for diagnostic test kit development. And the market prospect for India are huge. Enrich Bioscience platform is capable of diagnostic screening, therapy selection, and relapse detection. Our first product is for detecting cancer relapse because it provides a comparative analysis against the current testing method, and it is also the fastest route to go to market. The projection for three cancers for relapse detection in Indian market accounts to more than 100 million US dollars. And plus, we don't have to forget about the US market share serving the South Asian Indian immigrants over there, which are based on our model. So far, we have proved that our method is sensitive, accurate, and cost effective. And for that, we have already filed the patent. In a retrospective study, we demonstrated that the cancer signature detected in the cancer tissue and the blood sample were similar when measured from the same individual. Our next milestone is to validate NHS agnostic test in a real world scenario, and it is a prerequisite for a market launch. The plant study aims to compare Enrich Biosciences results with the routinely used clinical tests such as imaging, blood test, colonoscopy for relapse detection. 1,800 patients will be recruited. We plan to investigate three cancer types at two hospitals. Three cancers are lung cancer, renal, and colorectal cancer. And in this study, the patient blood will be drawn 6 months post surgery and tested by the Enrich Bioscience for result generation. The patients will be followed for another 12 months with routine clinical care. Um, and all the At 18 months, all patients will be assessed for their cancer status. And the results of this study will inform about two critical things. First, the number of relapse cases and which bioscience test could predict, and how sooner can we predict compared to the standard clinical examination. And with these results, NH will file the regulatory approvals. The study is divided into three phases: planning, that covers approval, setup, the workflow, and ongoing patient recruitment. And the second phase covers training cancer models on 300 patient samples for three cancers. While the last is the validation phase, and it is to test these models on 1800 patients as described in the study plan. And we are also very pleased to inform you that our funding round is open and we welcome any investment equal and above 250,000. NHS test falls under the in vitro diagnostic test kit category, and in India it is authorized by Central Drugs Standard Control Organization. Our collaborator, Tata Medical Center, is one of the testing sites for new kits before their market launch in India. NH test is offered at the certified diagnostic labs of the hospital and community testing units. To summarize, we have a cutting edge technology for sensitive, accurate, and affordable cancer testing. An NH Bioscience mission is to provide an accurate cancer test that is built by and for your community. Connect with us to learn more about NH Bioscience and how you can be a part of it. And I just want to mention this last slide that NH Bioscience mission is to offer you with convenient, early, and accurate detection tests for you and your loved one. And these are our partners, OptaScan. This is a cloud-based platform connecting two pathologists on a cloud for second opinion on histopathological slides. And the other one is Excellent Diagnostic. This is a breath analyzer for detecting infectious diseases. And with that, I thank you for listening to us and happy to take any questions. Thank you.
Well, thanks, Tarang. It's a terrific presentation and obviously a really serious problem. And it's just one of those biases that happen in the world. And it's great that you're taking this challenge on. Who has any questions for or suggestions for Tarang? Go ahead, Snita.
I was pausing because I noticed I was going first every time, so I wanted to give the others a chance. Tarang, thank you for a very eye-opening presentation. Not a problem that I was aware of, so that was very, really appreciate having my eyes opened here. A bit of feedback on the good side. It was the Mona Lisa example you used there was very impactful. I think that was able to take a presentation that's otherwise very technical and kind of help somebody who doesn't understand the space very well really appreciate the gravity of the problem. So just a piece of a compliment there on how you told the story and including that example in the presentation.
Thank you. Yeah.
Dr. Patel, you're muted. You're muted.
Yeah, I'm good. Okay, thanks. So yeah, again, great presentation. Really appreciate it. I think some of the detail wasn't quite clear to me. I actually work in sort of the cancer immunotherapy space. Our company develops cancer vaccines, and I know from my sort of very limited knowledge that the epitopes change or are not well defined for a lot of cancers. So how do you do that? How do you get the sensitivity and the specificity for these tests? And I guess the second question is, what's your sort of IP? What's your barrier to entry? Because clearly, Roche or anybody else, one of the big diagnostic makers, would be able to sort of look at this data and develop tests fairly easily to detect these types of variances.
Thank you for your question. It's a great question. To your first part, epitopes. So for that epitope thing is more on protein side. What we are looking are the DNA, the genetic material, and on the genetic material, we all know that ancestry has a biggest role. I mean, we can easily outlie the groups, ethnic group, just based on the DNA information. Said that, yes, the more I'm looking on various diseases, not only on cancer, I'm finding more and more biases, more geared towards the European descendant. And it's not that the companies or the academic researchers, they did it by intention. No, because yes, in North America, majorities are coming from European descendants,
yes.
Right? So that is one thing. And now with the growing literature, we are getting this sort of the understanding that ethnicity, the genetic material, is playing a major role, is one of the major variable in cancer modeling or other human disease modeling. To your second question about the Roche, Illumina, Grail, and there are so many other companies, they are mostly in North America. Definitely they can come to India, they can use the data, but unfortunately there's no data. And that's the reason we are partnering with AIMS, Tata Medical Center, and all the big hospitals, and as well as the academic researchers such as Dr. Satyamurthy, who is doing or generating this data. And our objective is to compare whatever is publicly available data from the North, let's say European descendant, or let's say American Africans or East Asians, and compared with the South Asian population, and then see what are the common things and what are the difference, and are there really a differences. In either way, we are gaining something. And if Roche or Illumina, they want to market their product in the Indian market, do it. There are other thousands other problems that we can easily work on. But that is not our objective. Our objective is to partner with them.
Mhm. Okay, no, that makes a lot more sense.
Yeah, I'm sorry for my previous comment, but it's a sort of upsetting that these sort of the research are not happening in India or in other developing or emerging countries.
No, I agree with you. It's very, very important to get this data out there. It certainly is. It's a very worthwhile project that you're doing. Definitely.
Thank you.
I have a question.
Sure.
So I
Sure,
love this
go ahead.
presentation, and like Sneeta, I wasn't aware of the 20% difference in the accuracy, and that's definitely worthy of investing your time towards developing this. So in terms of your product development, where is it at now, and where does it need to go over the next year? I saw the timeline you had for 6 months with 1.5 or 1 million. Can you get a bit more granular in terms of your product development plan?
So all the partnerships are in place. I mean, and project development, the project study is also being drafted out. We are already engaging with all the regulatory approvals, such as ethics approval, and negotiating what are the price point, and also looking for the vendors. This is on one side. Then the next stage is we are waiting for COVID to be over before we start the patient recruitment. And our first challenge is to have sufficient number of patient samples for each category to be recruited before we start doing any experiment and start to do modeling on the cancer. So yes, we are waiting for COVID, and hopefully by the end of this year, COVID will be over and the cancer patient will be start coming back to these hospitals. That's the biggest challenge right now we have. Other than that, yes, funding is another problem right now we are facing. We are getting small fundings, but that is just sufficient to pay to the technician to collect the samples.
Thank you. And like Sneeta
Well,
said in
thank
the
you.
presentation, sorry, I would focus on really clarifying the metrics that you want to emphasize. The story is good and analogies, and make sure that the key points are very visible to the audience who needs to see exactly what are the takeaway numbers that we need to see leaving this presentation.
Okay, thank you. Thank you, guys. Thank you.
Well, thank you very much.